Hello students. So today we are going to do the second part of the chapter biotechnology. In the previous part we learned the introduction to biotechnology, the traditional and modern biotechnology, rDNA technology, PCR, that is polymerase chain reaction. Also we learned the mechanism of PCR, the biological tools, restriction enzyme, recognition sequence, cloning vectors, that is plasmid vectors for plants, competent host, right? So today in the second part we are going to learn about the further methodology of rDNA technology, that is how insertion of DNA foreign gene into a cloning vector and expression of gene to obtain a desired product. Also we learn about healthcare biotechnology, which you can see in the present slide: vaccination production, gene therapy, agriculture, again the different forms of gene therapy such as germline gene therapy, somatic cell gene therapy. Right now, starting with today's part, as we can see on the present slide, the biotechnology in the healthcare sector. Like you can see how the diagnosis with the present day technology it can be done, whether it is related to the heart functioning or CT scan, or related to observe the inner part of the lungs, or the entire body imaging can be observed with the help of the modern technology. Further, you can see the second slide where a bacterium, in it bacterial chromosome is present as well as a plasmid. Cell containing gene of interest is removed and it is inserted into the plasmid, whereby we can make new gene by insertion or by using the restriction enzyme, as we learned the biological tool, the scissor, then the sticky ends which are present can be joined by the ligase, this DNA ligase enzyme, which acts as the gum or the glue to join the strand to form a new DNA strand. Right, so that forms the recombinant DNA plasmid. So that is how the recombinant DNA process takes place in simple ways. Further, now methodology of rDNA technology, where the steps involved in gene cloning are as follows. The step is isolation of DNA, that is gene, from the donor organism, that is the one which you want to isolate and clone it. So first point, the desired gene to be cloned has to be obtained from the source organism, or as we call as a donor organism, the from one which we are to obtain. Initially the cells of donor organism are sheared with the blender and treated with suitable detergent. Gene material from the donor is removed, isolated and purified by using several techniques. Isolated DNA can be spooled onto a glass rod. So by various methodology we can remove it, shear it, blend it, treat it with a particular detergent which is specifically used while isolation of DNA. Secondly, isolated pure DNA is then cleaved, means it is cut by using a restriction enzyme, as I mentioned, the biological scissor, particularly restriction endonuclease, that is RE. These enzymes cleave DNA at specific sites, means wherever you want to make a cut, called restriction site, and break the DNA into fragments. There are several types of restriction endonucleases. Cleaved DNA fragments have cohesive, sticky, staggered ends or blunt ends, means the one region where we cut or cleave the DNA has got a sticky end. From cleaved DNA fragment, a fragment containing desired gene is isolated and selected for cloning. So the part which we had cut, from that fragment we can remove the desired DNA. This is now called as foreign DNA or passenger DNA. A desired DNA or gene can also be obtained directly from genomic library or cDNA library. Now the genomic library or cDNA library where already the desired gene is stored or is readily available. Second, insertion of desired foreign gene into a cloning vector or vehicle DNA from which we can multiply or acquire the particular gene of interest by cloning. The foreign DNA or the passenger DNA is now inserted into a cloning vector or vehicle DNA. The most commonly used cloning vector are plasmids of bacteria and bacteriophage virus like the lambdaphage and M13. The most commonly one used is plasmid PBr322, as we have learned in the previous lecture or previous part of this chapter. Plasmids are isolated from the vector organism, that is a bacterium. Just now as we saw the previous slide where from the bacterium how the plasmid can be used, where the desired gene is inserted and cloning can be carried out. By using same RE, that is restriction enzyme or endonuclease, that is vector DNA is cleaved, that is it is cut. Further, now by using enzyme DNA ligase, foreign DNA is inserted into vector DNA. The combination of vector DNA and foreign DNA is now called recombinant DNA or chimeric DNA, and the technology is referred to as rDNA technology. Just as I mentioned in the beginning of the slide, as we saw the bacterium from which plasmid is taken or the vehicle vector in which the desired gene is inserted and the cloning is carried out, which the entire technology which is called as a rDNA technology. Third part, transfer of rDNA into suitable competent host or cloning organism. Now further it is introduced or transferred into a competent host or a cloning organism. Finally, the recombinant DNA is now introduced, that is transferred for expression into a competent host cell of the suitable cloning organism, which is usually a bacterium. Again referring to the second slide which you observed. Host cell takes up naked rDNA by the process of transformation and incorporates into its own chromosomal DNA, which finally expresses the trait controlled by passenger DNA. The transfer of rDNA into bacterial cell is assisted by divalent, that is calcium ions. The cloning organism used in plant biotechnology are E. coli, that is Escherichia coli, and Agrobacterium tumefaciens, which are most commonly used organism in case of plant biotechnology. Okay. Further, the host cell which has taken up rDNA is now called transformed cell. Why? Because it gets transformed by inserting the new DNA or the gene. Foreign DNA can also be transferred directly into the naked cell or protoplast of the competent host cell without using a vector. So here it can be transferred even directly into the protoplast of the particular host cell. This is done by using techniques like electroporation or microinjection or liposuction, shotgun, ultrasonification, ballistic method. So these are the different techniques by which we can even introduce directly into the protoplast of the host cell rather than using a particular bacterium. Okay. But in plant biotechnology, the transformation is through Ti plasmids of A. tumefaciens. That's just now as we learned Agrobacterium tumefaciens, which is used for the transformation specifically in plant biotechnology. Further, selection of transformed host cell. So now we are going to select the particular host cell. The transformation process generates a mixed population of transformed and non-transformed host cell for isolation of recombinant cell from non-recombinant cell. Marker gene of plasmid vector is employed. For example, PBr322 plasmid vector contains different marker gene, ampicillin resistant gene and tetracycline resistant gene. So here a particular marker gene is used. Further, when PST1RE is used, it knocks out ampicillin resistant gene from the plasmid so that recombinant cell becomes sensitive to ampicillin. That's where the PS1, that is RE restriction endonuclease, is used. Further, multiplication of transformed host cell. Now when you say multiplication, we can use a simple word of cloning or making more copies. Once the transformed host cell are separated by screening process, in this step, transformed host cells are introduced into fresh culture media. So when you're using into a fresh culture media where the multiplication or increasing the number of cells or making the copies is done or carried out. At this stage, the host cell divide and redivide along with the replication of recombinant DNA carried by them. As we said, multiplication or making more copies is carried out. Further, expression of gene to obtain the desired product. The next step involves production of desired product. So once you obtain the copies or the number of replication of the recombinant DNA, now we can obtain the production of the particular or desired product like alcohol, enzymes, antibiotics, etc. Finally, the desired product is separated and purified through downstream process, again by using a suitable bioreactor. So on to the right side, you can see the image donor cell, vector, that is a plasmid or a bacteriophage. The isolation of the desired DNA or target DNA is obtained, and then at the same time the vector or vector DNA by enzymatic fragmentation. By using restriction enzyme, the cut is made in the plasmid and the target DNA is inserted into it. That is by joining target DNA and cloning vector by DNA ligase. As I mentioned, what is DNA ligase? The glue or gum which helps to join the plasmid and the target DNA or the specific DNA. Then further, we can see the introduction into a bacterial host cell, this transformation of the viral infection. Just now as you saw the step-by-step process, here now this particular plasmid is introduced into a host bacteria, or which we use the term transformation. So transformed host cell containing rDNA molecule, further identification and selection, amplification, further cell multiplication, protein production from clone gene, then protein encoded by insert DNA. Further, we can obtain protein as the term protein harvesting. So this is just an outline process of recombinant DNA technology, or how it can be done systematically, step-by-step process. Further, again, just general information, that is, do you know gene library is a collection of different DNA sequences from an organism where each sequence has been cloned into a vector for ease of purification, storage, analysis. There are two types of gene libraries on the basis of source of DNA. Now, as you know, library where information, resource, everything is available. Similarly, gene library where again this collection of different DNA sequence can be obtained. Based on that, the two types, genomic library and cDNA library. Genomic library, it is a collection of clones that represent the complete genome of an organism. The genomic library of prokaryotes can be constructed by using plasmid vector. So if you want prokaryotes, we can use plasmid vector for the construction of these prokaryotic types. It is because prokaryotic genome does not contain any repetitive sequence, so it becomes easier to make a clone. cDNA library, the second type, it represents library of eukaryotic organism only. So in case of C, that is complementary DNA library where only it is for eukaryotic, where if you see in genomic, it can be used for prokaryotic. Further, DNA is produced from isolated mRNA by reverse transcription. The DNA so made is called as complementary DNA, or in short we learn as cDNA or complementary DNA. The library is called cDNA library. Eukaryotic DNA genome contains introns, regulatory gene and repetitive DNA. Hence the establishment of genomic library in eukaryotes is not meaningful. Now you can see the image of genomic library and cDNA library, or how it is made. Again, with the help of the chart which I have provided over here, which helps to understand the difference in genomic library and cDNA library. First you can observe the genomic DNA library, how the process takes place. You can see the genomic DNA, the isolation of DNA from these cells, and further along with that simultaneously plasmid vectors, the digestion with the restriction enzyme, that is cutting the particular site in plasmid DNA, as well as the cleaved DNA, which is again joined by using DNA ligase, that is the glue, and then it is again inserted into a bacteria for amplification of the genomic library, and then the collection of genomic library by DNA isolation. At the same time, if you see cDNA library, where mRNA, that is by reverse transcription, you get reverse transcriptase cDNA. Now again, at the same time, simultaneously the vectors are taken, where cleaved DNA, digestion with the restriction enzyme, and it is joined with the cleaved vectors, that is by ligation, and then we can insert it into the E. coli, where library amplification is rapidly obtained, that is reproduction of the bacteria, and again you can do the collection of complementary library of the actively transcribed genes. So you can see the small difference between a genomic library and a cDNA library. Now, so further little bit of additional information over here, which is given in the textbook. CCMB, or the Center for Cellular and Molecular Biology, located in Hyderabad, is a premier research organization in frontier areas of modern biotechnology, including DNA, fingerprinting and molecular approaches in animal breeding is carried out. The main objective of this center is to conduct high quality basic research and training in frontier areas of modern biotechnology, and it also helps to promote centralized national facilities for new and modern techniques in interdisciplinary areas of biology, that is in different fields of biology. Towards the right you can see the entire CCMB, that is the Center for Cellular and Molecular Biology Institute, which is located in Hyderabad. Now further application of biotechnology. Now from the previous one where we learned about how the methodology is carried out, now here we can see application where it is applied. Now again coming to the very first slide where I showed you the how the diagnostic can be carried out in the present day method, where with the help of biotechnology or latest technology we can scan the entire body, do CT scanning, imaging, etc. So here we can see application of biotechnology. Biotechnology is an umbrella term covering a broad sector of scientific application used in many sectors such as health, agriculture, industry, environment and genomics. So it is as a term, umbrella term means it is a spread out part where we'll not only learn any one or two aspect but more number of aspects, such as animal biotechnology, where tissue culture, generative medicine, immunotechnology, GM, that is genetically modified animals, pharmacogenomics and gene therapy. Also environmental biology, where pollution control, biofuel, bioremediation and biodiversity is carried out. Third, microbial biotechnology, where microbial genomics, biofertilizers, biopesticides, IPM, whereas genomics, whole genome sequencing, gene function, reverse genetics, bioinformatics. Industrial biotechnology, enzyme biotechnology, protein engineering, metabolic engineering, metabolomics, whereas Plant biotechnology, plant cell and tissue culture, again GM crops like GM animals, marker assisted selection. So these are some of the applications of biotechnology. Okay. Now, I added few more images to make you understand how and what are the different technologies used and which helps us in the present scenario, that is medical biotechnology, diagnostic, therapeutics, vaccines. Again, coming to the first slide where you saw how the diagnostic can be carried out. Agricultural biotechnology for plant agriculture, farms and food processing, which is again a very important aspect in present day situation. Then environmental biotechnology, cleaning through bioremediation, preventing environmental problems, forensic biotechnology, paternity tests, scientific investigation, industrial biotechnology, energy productions and new materials. Okay. Now we take one by one of all the applications. First, we come across the healthcare biotechnology. So that is why I emphasized on the first slide with the healthcare biotechnology or diagnostic product. Now, it refers to a medicinal or diagnostic product or even a vaccine. As you know, presently the vaccine for COVID-19 is in process, right? There are few vaccines already made, but this again comes under the healthcare biotechnology. This technology has a tremendous impact on meeting the needs of patients. Biotechnology offers patients a variety of new solutions such as unique, targeted and personalized therapeutic and diagnostic solutions for organ transplantation, stem cell technology, genetic counseling, forensic medicine, gene probe, genetic fingerprinting, and karyotyping are outcomes of biotechnology. So if you see the image or the animation which I kept, which you can somewhat relate with the stem cell technology, or even with the organ transplantation, etc., where it helps us to understand the different organs or the stem cell technology which can be carried out. Further, human insulin. Now, as we know, the human insulin, which is very much important for the diabetic patients with type 2. Insulin is a peptide hormone produced by beta cells of islet of Langerhans of pancreas. It was discovered by Edward Sharpe Schaeffer in the year 1916 while studying islet of Langerhans. Insulin is essential for control of blood sugar levels. Diabetes mellitus is a disease in which some people cannot make insulin themselves, as diabetes mellitus means high blood sugar or more amount of sugar present in the blood. So for this condition, insulin becomes very important for the persons to control the blood sugar. Otherwise, it will rapidly increase in the human body, thereby causing harm to the various organs like eyes, heart, kidney, etc. So that is why in case of diabetic mellitus, insulin becomes very much important. Further, Hakura et al. 1977 chemically synthesized DNA sequence of insulin of two chain A and B and separately inserted into two PBR 322 plasmid vector, as we just learned most commonly used plasmid. Insulin production by recombinant DNA technology is designed by Gilbert and Vinokourov in 1978. Further, the genes are inserted by side of beta-galactosidase gene of plasmid. The recombinant plasmid were then separately transformed into E. coli hosts. The hosts produced penicillinase pre-proinsulin. Insulin is later separated by trypsin treatment. Where you can see on the right side, again I provided an image where you can see how it can be produced or how the production takes place. Human pancreatic cell, the DNA, human insulin producing gene, bacterial DNA, plasmid DNA, from where plasmid DNA is cut with the restriction enzyme, that is the scissor, biological tool. Then recombinant DNA is inserted, joined, that is by the help of ligase. Introduction of recombinant DNA into the bacterial cell or the host cell. Further, the recombinant bacterium multiplying and producing human insulin in the fermented tank. And you can see how the insulin extraction and purification is taken place, or production of human insulin is carried out. Okay, here again I put a chart which is there in the book, showing human proteins produced by rDNA technology to treat human disease. Disorder, anemia. What is the recombinant protein used? Erythropoietin. Asthma, interleukin 1 receptor, atherosclerosis, platelet derived growth factor, parturition, relaxin, blood clot, tissue plasminogen activated TPA, urokinase. For cancer, interferon, tumor necrosis, factor interleukins, macrophage activating factor. Diabetes, insulin, emphysema, alpha 1, antitrypsin, hemophilia A, factor 8, hemophilia B, factor 9, hepatitis B, that is hepatitis B vaccine. Again, I put an image showing how the treatment of disease takes place. Recombinant DNA technology can be used to make important proteins that could prolong and even save human lives. For example, human growth hormone, which is used to treat patients suffering from pituitary dwarfism, that is short-sightedness, is now widely available because it is mass produced by recombinant bacteria. Other products now made in genetically engineered bacteria include insulin to treat diabetes, which is now very common among people because of the food habits or stress, strain, etc. Blood clotting factor for hemophilics, and potential cars cancer fighting molecules such as interleukin 2 and interferon, because again cancer, it can be of any part, any organ, etc., and it is again more found in the in many people because of various reasons, right? So you can see the image how showing pituitary gland, liver, bone, adipose tissue, skeletal muscle, and again how it can be produced in case of diabetic, where pancreatic cell produces the insulin. Further, vaccine production. Now if you see on the right side, you first see the image. Now in the image you can see how the vaccine production is carried out in a large scale. Again, if I point out present day situation for corona, the vaccines has to be produced in lakhs and lakhs, or we can say in millions, because for example in India itself we have got 137 million population, right? So in that case the production should be in larger number. So in that case, if you see how the production takes place in a larger number, not only for people in India, but is also exported to other countries also, right? So we can see how the production is taken place in large scale. So coming to the points, a vaccine is a biological preparation that provides acquired immunity against a certain disease. Usually, a vaccine consists of a biological agent that represents the disease-causing microorganism. It is often made from a weakened or killed form of the microorganism, its toxin, or one of its surface protein antigen. What does that mean? The vaccine is produced for a particular disease which is obtained from the disease-causing microorganism itself, which may be a killed from that particular microorganism, or toxin taken from it, or the protein antigens from that particular microorganism, and then the vaccine is made. Second point, biotechnology has offered modern diagnostic test kits, rickettsial, bacterial, viral machines, along with radio-labeled biological therapeutics for imaging and analysis. Vaccines have eliminated smallpox, polio, and other deadly disease for the last several decades. That's why if you see nowadays the cases of smallpox, which is already eradicated, polio is also nil, and even several other viral diseases which have been stopped or eradicated because of the vaccine, if it is taken on time during the childhood or from 0 to 5 years, the vaccine which is given to the children's. Further, biotechnology has made advancement in vaccination by making recombinant vaccines that have the potential to eradicate non-communicable disease like cancer. Naked DNA vaccines, viral vector vaccines, and plant-derived vaccines are found to be most effective against a number of bacterial and viral disorders. Moving further, oral vaccines a novel approach. Just as I told, the as a child is born from 0 to 5 years, various vaccines are given like DTP, hepatitis B, HIV, HPV, MMR, pneumococcal, polio, rotavirus. So we have to always keep updated, check with the doctor what all vaccines you have to take at what month, what which year, on time, every time, right? So we have to keep updated with the various vaccination which has to be given to the children's, mainly from 0 to 5 years, so that we can ensure that our family is protected, the child is protected against viral and bacterial disease. So we will go to the points one by one. The latest hotspot in the field of vaccine research is the development of vaccine which can be taken orally, like polio vaccine, right? Immunogenic protein of certain pathogens is found to be active when administered orally. The gene corresponding to such protein is isolated and a gene construct is produced. This is introduced and expressed in a plant genome, which results in production of such immunogenic proteins in the parts of plant where it is expressed. These, when fed into animals or mainly humans, the person becomes vaccinated against certain pathogen. Such vaccines are known as edible vaccine. That is why the name given as oral vaccine, because oral we take it through our mouth region, where that's why the name given as edible vaccine. Okay, as we consume food, correct? Then an exciting invention is production of melt in the mouth vaccine that can be administered by placing them under your tongue, that delivers it into the bloodstream. Now it is very easy to understand, that is a vaccine which is kept under the tongue, it melts and directly enters into your bloodstream. Okay. Further, the most important example is the production of flu vaccine by bacillus, which melts in the mouth. The tremendous benefits of such vaccines is the comfort of administration, low cost, and easy of storage. Now, as you say, a very simple words, you can prevent injections, that is pricking onto your body. Secondly, low cost, the easily it can be administered, and also the storage becomes easier. Okay. Further, now from the healthcare we move to the agriculture sector. Again, if you see the image provided, it will make you understand how the use of drones, which can use for the spraying of pesticide, insecticide, or observing the entire field area, etc. And also the different machineries or tools which can use even without the use of man, or manually it can be operated with the help of technology. Applications in agriculture: pest resistant plant act as biopesticide, it reduces the need for insecticide. Example: bt cotton, bt corn, rice, tomato, potato, soybean, etc. So that is another image. Coming to the points, application of biotechnology in the agriculture involves scientific techniques such as genetically modified organism, bt cotton, pest resistant plants. It helps in modifying plants, animals, and microorganisms and improve their agricultural productivity. Tissue culture is used in micropropagation. The term micropropagation, that is by using small part of the plant, whether it is stem, root, leaf, it can be produced in large scale, as given large scale propagation of plants in a short duration or in a very small time period. Further, tissue culture technique is also the best method for storing germplasm and maintaining a specific genetic type, or we can say clone. This technique is used in those plants which produce recalcitrant seeds or produce highly variable seeds. Okay. Further, coming to third part, gene therapy. Again, see the image how the particular gene, particular DNA, particular part can be identified and the gene therapy can be carried out. Again, coming to the point, a gene is a stretch of DNA required to make a functional product such as part or all of the protein. During gene therapy, DNA that codes for a specific gene is delivered to individual cells in the human body. Most of, if not all, disease can be or have a genetic factor. The genetic factor can be wholly or partially responsible for the disease. For example, in disorders such as cystic fibrosis, hemophilia, and muscular dystrophy, changes in the gene direction results in the condition. Now, as we just read the 3 points, here we can say easily the gene which is been affected or the stretch of DNA where the particular gene is affected can be identified. It can be removed wholly or partly where which is responsible for disease, and it can be cured or changes can be made in that particular disease, as the given the example like cystic fibrosis, hemophilia, or even muscular dystrophy. Further, in other conditions such as high cholesterol and high blood pressure, genetic and environmental factors interact to cause disease. There are more than 5,000 different human genetic disease known to cause by single gene defect. So one single gene defect can cause almost 5,000 different genetic disease. Now here you can see the examples: sickle cell anemia, thalassemia, Tay-Sachs disease, cystic fibrosis, Huntington's cholera, hemophilia, alkopenia, and albinism, which are some of the diseases caused by a single gene defect. Further, gene therapy is being used in many ways. For example, just you can see the two images, one where you can see the particular gene which is affected or DNA strand can be replaced, or second where the animation where you can see the DNA with the particular mutated clone and no protein or the protein which can be altered. So you can see replace missing or defective genes, as you can see in the image, deliver genes that speed the destruction of cancer cells, supply genes that cause cancer cells to revert back to the normal cells, deliver bacterial or viral genes as a form of vaccination, as just we learned in the previous slide, deliver DNA to antigen expression and generation of immune response, supply of gene for impairing viral replication, means to stop the viral growth or to multiply from growing, then provide genes that promote or impede the growth of new tissue, deliver genes that stimulate the healing of damaged tissues. Clear? Now from the term itself it is very clearly understood what and how the gene therapy can be useful. Further,